Welcome to today's EPI-WIN webinar. We welcome you all, particularly those of you joining from far away or late or early times. I know many of you have joined when it's really, really early, so we really thank you for joining. So today we will have our webinar on the public health risk of avian influenza A H5N1 detected recently in dairy cattle. We have a great panel. It will be moderated by Dr. Wenqing Zhang, who's the head of the Global Influenza Program here at WHO. But before we go to that, I'd like to announce some housekeeping. Those of you who know EPI-WIN already are familiar with this, but this webinar is being live streamed on the EPI-WIN YouTube channel. It's also being recorded. And afterwards, the recording will be available from our website and will be shared for everyone who's attended this webinar. So don't worry. We do not have interpretation today, but we will have the language transcript when we put it on our YouTube channel. So it will be available in other languages later as well. We invite you all to post your name, location, and organization in the chat box. We'd love to hear from you and where you're from. And please if you have any questions, add them to the Q&A box, not in the chat, because that's how we can keep track of the questions and make sure they're answered. So your name, location, and organization in the chat box, but your questions in the Q&A box. So without any further ado, I now hand over to Dr. Wenqing Zhang. Over to you, Wenqing.
Excellent. Thank you very much, Supriya. I hope you can hear me well. Okay, great. So welcome everybody to the WHO EPI-WIN webinar on public health risk of avian influenza H5N1 detected recently in dairy cow. The title is self-explanatory. So without further ado, let's welcome Dr. Maria van Kerkhove, Acting Director of the Department of Epidemic and Pandemic Preparedness and Prevention of WHO, to give her opening remarks. Maria.
Thanks, Wenqing, and thanks to the EPI-WIN platform for giving us this opportunity to give you an update on H5N1 from a public health point of view. So I think the first thing I just want to mention is WHO's work on influenza spans more than 70 years, and you'll hear a little bit about that today, starting with the Global Influenza Surveillance and Response System. And together with PIP, which is our Pandemic Influenza Preparedness Framework, which came into existence in 2011, we have a large portfolio of work in this space, and we work with many partners around the world, some of whom are on this panel today. What we're trying to do through looking at influenza viruses that are doing regular ongoing risk assessments of looking at this virus that's in circulation. And what we're focused on here today is H5N1, but there are other avian influenzas. And of course, you know about seasonal influenza and the threat and risk of pandemic influenza. But I do want to stress that WHO doesn't work alone. The Global Influenza Surveillance and Response System has partners of more than 150 labs in more than 130 countries. We work across the animal sector with WOAH, with FAO, not just at an international level, but at national levels, we work with animal sector partners as well. And what you're seeing as this unfolds with H5N1 is One Health in action. You cannot look at human health risk without looking at the risk in animals, looking at the risk of the animal-human interface, and us working together to articulate what this risk is and how it is changing really needs our partners to be able to do the work in real time, share this information in real time so risk assessments can be done in real time. Right now, there's a lot of focus on the US, but we are seeing a global epizootic of avian influenza. And we've seen H5N1 infected in wild birds and poultry and marine mammals and land mammals. But what is concerning is that we are seeing new species that are being infected. And you'll see this being presented today and you've heard about in dairy cattle. And so for us at WHO, what we're trying to understand is the exposure risk to humans. From a public health perspective. And we will hear from colleagues on the animal sector as well, looking at risk in milk, looking at risk in mink, and looking at risk to the herds themselves. And what we are trying to do at WHO is put out advice around to the general public, but really importantly to those who are occupationally exposed to infected animals. And for us, we need much stronger surveillance in animals globally, not just in the US, looking at the species that we know can be infected with H5N1, but also in humans at the animal-human interface. And again, here I'll mention the occupationally exposed individuals, because prevention is key. Those that are doing surveillance, we need you to continue to sequence. We need you to continue to share those sequences so regular assessments of the viruses themselves can be analyzed, as well as understanding what any changes in these viruses mean in terms of transmissibility, in terms of severity and whatnot. But again, prevention is key in the animal sector, between animals to humans. And we also, within this webinar, we'll probably get some questions on this, is around preparedness for vaccine production. With the GISRIS and with PIP, we have a vaccine pipeline for influenzas. I want to stress that we're not in a situation where that needs to be triggered, but we do want to reiterate that there is a system that is in place to be able to increase production for H5N1 should we need. And there are some candidate vaccine viruses that are available to manufacturers should they need to ramp up production. Again, we can get to that later, but I did want to stress that we're not at that point. So our relationships are key. Thank you so much to all the panelists who are here. Thank you to all of the members of GISRIS across the animal sector within food safety. And we're really grateful to have our food safety colleagues online today to discuss the risks in meat and in milk. And we can only do what we do with those partnerships. So big thanks to this, not just at a global level, but also at a regional level. And with particular focus on the US today, I also want to say thanks to PAHO for their continued work on surveillance for influenza, but even broader for pathogens with epidemic and pandemic potential. So I wish you a very successful webinar, and we look forward to answering your questions. And back to you, Wenqing.
Thank you very much, Maria, for introducing the background. So with this, we can move to the next part of this webinar. It will be a presentation shared by two WHO experts, Dr. Aspen Hammond from the Global Influenza Program and Dr. Moez, Sanaa, the unit head of the Standard and Scientific Advice on Food and Nutrition. After the presentation, we will have Q&A session, and we have a very strong panelist presented. We'll introduce them later. So let's kick off the presentation. I give the floor to Aspen.
Thank you, Wenqing, and thanks to the panelists, and thanks for everybody joining and their interest in this topic. Next slide, please. I am just going to give a very brief overview of the situation. It's a very rapidly evolving situation, so information changes quite quickly, but myself and a colleague from our food safety department will give this presentation to set the context a bit before the question and answer with the panelist session that will follow. So as most of you probably have read or heard, in early 2024, earlier this year, there was an unexplained illness in dairy cattle that caused a drop in their milk production. And this was investigated, and it was actually the detection of H5N1 in the milk from the cows that was reported on the 25th of March that began this series of events. One of the other important milestones in this event was the detection of H5N1 virus in a human case, somebody who had conjunctivitis and had direct exposure to dairy cattle. And that infection was notified to WHO soon after detection, so that was the beginning of April. Other important events that have happened in this situation include the detection of highly pathogenic avian influenza in pasteurized milk, and a lot of further studies are underway on food safety in this regard that we'll hear about a little bit later. So as of a few days ago, there were 36 dairy cattle herds in the United States among 9 states that were affected. There have also been detections of H5N1 in other animals near the affected dairy cattle herds. So we've heard reports of infections detected in cats and raccoons and wild and domestic birds nearby. When WHO received this information initially, and as more information came in, we did a joint situation assessment with our animal health partners, the Food and Agriculture Organization of the United Nations, and also the World Organization for Animal Health. And we looked at the risk assessment, the preliminary risk assessment. And the WHO assesses the overall public health risk posed by H5N1 to be low. But for those with exposure to infected birds or animals or other contaminated environments, that the risk of infection is low to moderate. But we stress that this risk assessment requires close monitoring, and WHO and our partners will continue to regularly assess and publish updated risk assessments for H5N1 and other avian influenza viruses. Next slide, please. So this just, again, going back to the detections in dairy cattle in the United States, you can see a map. The USDA updates this map with the states that are affected, and just a timeline on the left of the confirmation of H5N1 in herds, again, that began towards the end of March. Next slide, please. So just a few more details about the detection in cows. It's reported that in the milk from affected cows, there's a very high viral load of H5N1, and that's in contrast to a low viral load coming from swabs, nasal swabs collected from these cows. There have also been reports that asymptomatic cows have tested positive for H5N1, and that also H5N1 was detected in the lungs of a cow from a slaughterhouse, from a cow that had no symptoms. It's also been identified that there's been spread between cows within the same herd and spread between dairies associated with cattle movements within the United States. It's suspected that from cow to cow, the transmission likely happens through mechanical means. But again, this is one of the areas that's under investigation, with a lot of work going on to understand how the virus is transmitted to and from and between cows. And previously to this, influenza A was thought to occur on a very infrequent basis in cows. So again, there's a lot of work needed to understand the role of cows and the transmission dynamics in this species. And as of the sort of end of April, the 24th of April, the USDA has put in some mandatory testing requirements for livestock and dairy cattle, especially to try to understand and limit the spread of this. Next slide, please. And regarding the detection in a human, the human case that I said was notified to WHO at the beginning of April. This is only one, there's been only one human case associated with this event. So there was a person who had conjunctivitis. This person had direct contact with cattle, presumed to be infected with H5N1. This person had onset of symptoms in late March. And the confirmation was done at the United States Centers for Disease Control and Prevention, late March. And again, no additional cases have been detected associated with the H5N1 virus in cattle thus far. And there is active monitoring of exposed persons and monitoring of influenza in the community using routine surveillance systems. Next slide, please. And just this is briefly an update on the characteristics of the H5N1 viruses. So from cattle and from the human case and from birds. So recent viruses, these remain primarily avian influenza viruses. There have been no changes detected in the virus receptor binding that would indicate a change in transmission to and among people. In terms of the genetic analysis, these H5N1 viruses belong to the H5 clade 2344B, the genotype B3.13, which has not been detected outside the US thus far. There have been the one mutation in the PB2 gene detected in the virus from the human case, and also in one virus from a cow with a mixed population of this mutation. No known markers for reduced susceptibility to neuraminidase inhibitors, and these are antiviral medications, or endonuclease inhibitors, except in one virus with one change associated with reduced susceptibility. And in terms of antigenic analysis, data from the hemagglutination inhibition assays conducted at CDC showed that the human virus was well covered by existing candidate vaccine viruses, or CVVs as they're referred to, and that the viruses from cows are also expected to be covered well by the CVVs, considering their almost identical genetic sequences. Next slide, please. And then just to step back and give a bit broader context around this event, so that we can understand, as Maria said, for over 70 years, there's the global influenza surveillance and response system has been in place, with the main mission to protect people from the threat of influenza, be it seasonal, zoonotic, or pandemic influenza. And there's a map on the screen that shows the location of a lot of the labs that are in this network. So they're national influenza centers, they are WHO collaborating centers, there are essential regulatory laboratories, and H5 reference laboratories. And next slide. And further on taking a bit of the perspective in the past, we have had over 800 detections of H5N1 virus in humans, reported to WHO from over 20 countries since 2003. More recently, a lot of the avian influenza H5N1 viruses belong to this clade 2344B, and they continue to diversify genetically and spread geographically. And since 2022, there have been a broader range of wild bird species affected globally, which has a lot of effects on the ecology of these birds and has caused a lot of mass die-offs in some species. And the situation in wild mammals is also worrying. There have been reports of some species suffering significant mortality events. So the situation is of concern. But despite the number of H5N1 outbreaks and detections in animals and the amount of human exposure to these viruses at the interface, relatively few human infections have been reported to date. So 28 human cases of H5N1 have been reported since 2021, all with people who had exposure, either direct or indirect contact with infected animals or contaminated environments. And among these cases, thus far, there's been no reported human-to-human transmission. And we also know that swine influenza viruses circulate in pigs in many countries, and we occasionally get reports of human infections with swine influenza viruses. And last but not least, we also see many other influenza subtypes that circulate in animals that are sporadically detected in humans as well, other than H5N1. Next slide. And just to, again, give a bit of a graphical overview of the detections in animals, these are maps courtesy of our partners at FAO. So the map on the right shows the period from 2022 to 2023. So you can see the detections of avian influenza in animals. And the map on the left shows the more current period from October 2023 through April of this year. So we continue to see these detections in many places around the world. Next slide, please. And as I said earlier, when we received this information, we looked very closely with our partners at FAO and the World Organization for Animal Health to do a preliminary assessment and look at the risk. And the risk, again, was unchanged. The overall public health risk was deemed to be low for H5N1, but for those who have exposure, the risk of infection is considered low to moderate. There's been no reported human-to-human transmission of H5N1 viruses since 2007. Prior to that, there were small clusters in humans involving healthcare workers, but no sustained human-to-human transmission reported. But again, this requires us to be vigilant and monitor the situation closely so that we can update our risk assessments. And next slide, please. So again, looking at the individuals who may be engaged in activities that involve exposure to infected animals or contaminated environments, they may be at higher risk for infection and should take necessary precautions to prevent infection. So as long as people are in contact with infected animals, now including dairy cattle, and if they do not have personal protective equipment, there may be a risk for human infection. However, the risk can be mitigated by measures to reduce exposure to the virus, such as using recommended personal protective equipment, implementing appropriate personal hygiene, and other risk-based biosecurity measures. Next slide. So I will just turn the floor over to my colleague from food safety to go over the next few slides on food safety aspects. Thank you.
Thank you, Aspen, for the updates. Yeah, good morning, good afternoon, good evening. So my presentation will discuss some food safety aspects. However, I will focus mainly on milk because we have more data on milk, on dairy milk. So first, next please. First, I would like to explain the process for raw milk versus pasteurized milk. As you can see in this slide, so you have the milking, cooling, the milk can be analyzed, and bottling. This is the process for raw milk. And for pasteurized milk, we have the same process. However, the milk from different farms are collected, analyzed at the dairy, unloaded at the dairy and refrigerated. And we have other steps, mainly the heat treatment. And we have cooling and bottling, conditioning, and sent to the market. This is the process of pasteurized milk. So why I remind that? Because the process is not only about heat treatment. We need to take into account all the good hygiene practices before we heat the treatment. And one of them is that in general, the cows are milked separately. Abnormal milk is redirected, not mixed with the other milk. And when we are collecting milk to the dairy plants, the milk from farms with zoonotic diseases are in general collected in special stream. So this is very important to understand. And in addition to that, the heat treatment, usually we define two conditions for heat treatment. The first one is at 63 degrees Celsius for 30 minutes, and the second at 72 degrees Celsius for 15 seconds. It is important to recall why we set those conditions. They are mainly set for highly pathogenic bacteria, and one of them is Coxiella burnetii. This setting should allow at least 5 log 10-step reduction of the initial load of the bacteria. So what does it mean? Does it mean for one single bacteria, the probability of survival is less than 10 to minus 5. That means we will reduce the 99.9999% of the bacteria present in milk. So this setting of pasteurization is mainly done for bacteria. We have some information for virus and not for H5N1. I will explain later what we know about H5N1. So as you can see here, we cannot see the pasteurization of milk only at the heat treatment step. So the importance of the good hygiene practice before heating the milk is to reduce as much as we can the virus or the pathogen load in the raw milk. Next, please. So what triggered the attention is that some sampling was made at the commercial level, and you see certainly this figure 1 out 5 or 20% of samples of commercially sold pasteurized milk tested positive on QRT PCR. So we have to be to make some caution with those figures. First of all, the sampling plan. I don't think that the sampling plan is representative to all the commercially sold pasteurized milk. We know in general in those situations, we target the sample. I think mainly the sample are coming from the area where the H5N1 is circulated, and especially milk produced from this area and for which the milk is coming from infected farms. So there is a question of representativity when we can say that the prevalence should be low. However, QRT-PCR doesn't mean that we have infectious live virus. So as of May 2, high number, I think that we are around 100 sample positive QRT-PCR was tested to check for the viability of the virus. So far, we don't have positive sample showing that we have live infectious virus in milk. So the available information today supports the argument that pasteurization effectively inactivates the H5N1 virus. Next, please. So pasteurization efficacy should be understood not only by observing what we have in the market, but we need to look at the two elements. First is the initial viral loads. And second, the survival capacity to heat treatment of this virus. For the second aspect, unfortunately, we don't have data on H5N1 in milk. This is the first time that we observe contamination of milk with this virus. However, we have data on eggs, meat, for which the main evidence that you have that H5N1 do not survive in eggs and in meat. So there is some ongoing studies to assess the resistance of H5N1 in milk, and we need as well to have an idea about the initial viral loads. So the preliminary results are encouraging. The virus is inactivated by heat treatment, conditions similar to milk pasteurization. However, we have only few experiments, so waiting for additional study with higher initial virus load and with varied other temperature of the heat treatment. So next, please. So when we look at the available data, I think we can say that the current pasteurization can be considered as providing the safety of drinking milk, because we are not looking only at the heat resistance of the bacteria, but we need to look at the current situation, because what is known that abnormal milk is not mixed with the normal milk. Secondly, for the moment, the majority of the farms recognized infected farms are not, their milk is not introduced in the market. So with that, we can continue this to repeat the same recommendation to the consumer. First, raw milk, eggs, or meat are not recommended. Not especially because of avian influenza, but for a variety of major pathogen and major foodborne diseases. So we need to make sure that pasteurized or you are consuming pasteurized milk. If there is no pasteurization, you can boil the milk, but boil the milk, you know how to do it to reach 100 degrees Celsius. And for the moment, we recommend mainly pasteurized milk products. However, we know that some technology that, for example, producing cheeses from raw milk are well advanced, and they can produce cheese in a safe way. So our recommendation that is to not consume raw milk products coming from non-certified company or recognized company that they are fulfilling the food safety standards. So this is our recommendation for the moment. And we will keep you informed with the new development when we have the more data about the resistance of the virus. But I'm not alarming, but the majority of the data coming and ongoing study showing that the current pasteurization condition should not present an issue for the consumer. Thank you, and I give the floor again to Aspen.
Thank you. Next slide, please. Thanks. Just back one, please. Thanks. I just wanted to conclude with a few more slides on some preliminary advice and some of the other activities that WHO is working on. So we are working on more detailed recommendations and guidance to minimize exposure and the risk of infection. But I just wanted to summarize in the next few slides some advice for people working on farms. So those at risk for exposure to potentially infected animals should be provided with and trained in proper use of appropriate PPE, personal protective equipment, which are exclusively dedicated to the work that they're doing, and wearing appropriate personal protective equipment based on the site-specific risk assessment, which could include gloves, coveralls, and face, eye, and foot protection, and respirator. And to regularly perform hand hygiene before and after contact with animals. And of course, if symptoms develop, that people should receive early and appropriate clinical management and be sampled according to the case definitions and investigations. Next slide, please. And for the general public, our advice, this is nothing different from what we've been saying before, but given the observed extent and frequency of avian influenza in poultry and wild birds and mammals, the public should avoid contact with mammals that are sick or dead from unknown causes, including wild animals, report dead birds and mammals, or request their removal by contacting veterinarians or local wildlife authorities. Of course, as we just heard, follow good safety and hygiene practices, properly handle and cook eggs, meat, other animal products, avoid slaughtering and preparing sick animals for consumption, avoid consuming raw milk, and again, seeking healthcare if someone is feeling unwell, and informing your healthcare provider if you have exposure, possible exposure to infected or potentially infected animals. Next slide. And just a bit to summarize some of the other activities WHO is working on in this area. So as we said, through the Global Influenza Surveillance and Response System, GISRS, this is a network of many influenza labs, especially collaborating centers who do a lot of detailed genetic and antigenic characterization of influenza viruses, looking at markers of mammalian adaptation, the genetic and antigenic similarity of viruses to the existing candidate vaccine viruses, looking at the virus's susceptibility to antiviral medications, validating diagnostic assays, trying to understand human population immunity to these viruses, and of course, using all this information to update risk assessments and update candidate vaccine viruses if there's a need. We also work on routine surveillance and monitoring for influenza and communications efforts such as this one, and to get guidance and recommendations out. And we also have a lot of ongoing preparedness activities, as mentioned, looking at the vaccine access through the Pandemic Influenza Preparedness Framework, clinical management, and pandemic surveillance guidance. And of course, working with our food safety colleagues on this event and other events. And of course, the international health regulations is important in this area. And we also spend a lot of time working with our partners, especially our animal health partners. Again, FAO, WOAH, and OFLU, which is the FAO WOAH Influenza Network, and other external partners. And next slide. So again, to summarize, it's important to understand the situation in animals globally. So countries should maintain surveillance in birds, monitor and investigate cases in non-avian species, so other including mammals, including livestock, report cases of detections to WOAH, so the World Organization for Animal Health, and other international organizations. We understand that a lot of people are working on transmission studies to understand better the role of asymptomatic animals in transmission, how the viruses are shed, where they replicate, etc., in different animal models, and doing serology studies in both humans and animals, and cows in this particular instance, validating diagnostics for humans and animals, testing of food products, as we discussed, maintaining surveillance in human populations so we can be vigilant and detect if something's changing. And of course, we encourage the sequencing and the sharing of genetic sequence data from both animal and human viruses, and look at, again, pipelines for vaccine development and deployment should it be needed. And we use all this information to update our risk assessments and provide updated guidance as the situation evolves. So I will conclude there, and I again thank everybody in the preparation of this presentation, and we look forward to having more discussion. Thank you.
Wonderful. Thank you very much, Aspen and Moez, for the presentation. Now, let me ask our panelists to please switch on your camera, and the speakers, please also stay on. And I would like to ask the panelists to do a round of self-introduction. Let's start with Todd.
Hi, everyone. My name is Todd Davis. I'm with the Influenza Division at the Center for Disease Control.
Thank you, Todd. And Mehdi?
Hi, everyone. I'm Mehdi Samayan, a technical officer at the WHO Global Influenza Program, responsible for zoonotic influenza and other laboratory-related matters.
Thank you, Mehdi. David?
Aspen, I'm a poultry veterinarian. I'm representing OFLU, the Animal Influenza Network.
Great. Last but not least, Richard.
Good morning, afternoon, evening all. I'm Richard Webby from the WHO Collaborating Center at St. Jude Children's Research Hospital, Memphis, US.
Wonderful. So we have a very strong panelist covering many areas relevant to this topic. I've seen a lot of questions. Some of the questions actually can be clustered together, so I'll be trying to do so. So let's kick off. Let's start with the first question, a question actually posted before our presentation even started. That's a question of our title, Public Health Risks. So what is the public health risks associated with H5N1 detected in dairy cattle? I would like to invite our two WHOCC colleagues, Todd and Richard, to address those. So Todd, if you can start.
Sure. Thanks, Wenqing. So the Center for Disease Control and Prevention still considers the risk to public health to be low. This is a virus that after sequencing now more than several hundred viruses from cattle, we don't see any molecular changes that would indicate increased possibilities of infection or transmission from person to person. So we still consider this public health risk to be quite low. I think some of the exceptions may be those that have prolonged unprotected exposure to infected dairy cattle. So I think there are some likely risks associated with occupational exposure. With that, I'll turn it over to Richard to comment more.
No, yeah, not much to add there. I think perhaps just highlight the fact that, this virus and its closely related genotypes have been, of course, very, very widespread over the past couple of years, not just in the Americas, but other parts of the world. And I think that the relative number of human cases detected over that period has been low as well. So it's pretty clear this virus is still primarily a bird virus that is poorly infectious in its current state for humans. Over.
Thank you very much. So let's move to the next question. And I would like to ask David, because a lot of questions really asking about the response in US. A lot of questions like what is likely the first infection occurred in cows, and what about the mechanical transmission looks like, and also about some figures, if you have, about the negative because the testing is going on, the negative number of detections in cattle. If you have these type of figures, would be great. So I give you the floor, David.
Thank you. So I'll just start off saying that just to make sure we all are on the same page, you know, the first cases that were diagnosed were in dairy cattle in Texas. This particular virus, as already mentioned by Maria, is a specific genotype. So it is the 2344B, but it has reassorted in North America with our low-path avian influenza viruses. So what we see in dairy cattle is the B3.13 genotype, which makes it, when you look at the 8 gene constellations, a virus that is not identical to what is circulating in other parts of the world. So in the risk assessment, that would need to be taken, that information need to be taken into account, that the risk may be different in other parts of the world because it's not the exact same 8 gene reassortment that we have in dairy cattle. The first case that was identified in humans, and there's been a couple of papers published, one in emerging infectious diseases. That was the first cases that were done postmortem wise, and the testing. The author is Burrow, and it's an emerging infectious disease that came out 2 weeks ago. And there's currently one on the preprint by again at State and the U.S. Department of Agriculture as a joint paper, and that has a lot more of the information about the genetics of the virus. So at this point, you know, the issue becomes transmission and how it appears from the studies by USDA, Iowa State, and that coalition, as well as some other ones that have been posted online, that it's most likely been a single introduction into dairy cattle. And from the genomic analysis, the phylogenetic analysis, it appears that it was a single introduction, and the moving of dairy cattle has spread that to multiple farms in different locations. The mechanism of the transmission between cows is currently unknown, but based upon field observations, it would suggest that the transmission probably occurs in the unique environment of a dairy parlor, either through a mechanical transmission through the milking process, or because there's continual cleaning going on in the dairy parlor, there are large droplets produced from that washing down process. So those are 2 leading theories or hypotheses, but they are not at this point proven hypotheses of how that transmission occurs. And the same thing is true of the one human infection. There are suggestions because of the conjunctivitis, it could be a transplant of the virus from contaminated hands of a milking worker, or it still could possibly be from the environment with the large droplets circulating possibly in that environment. Thank you.
So maybe I'll ask Todd if you could also add, because again, a lot of response, although so far we only have 1 confirmed human infection, but a lot of response has been taking place and is also ongoing on the US side. So would you be able to add a little more?
Yeah, thanks, Wenqing. So in the United States, when a confirmed dairy farm has been detected, and that detection is done by the U.S. Department of Agriculture, at the state level, the veterinarians will report those confirmed dairy farms to state public health departments. And then the state public health departments will take actions to monitor individuals on those farms where there have been confirmed dairy cattle. And so that can be in the form of active monitoring, where the names of workers are provided to local or state public health departments, and then those individuals can be monitored for up to 10 days post-exposure. And then there's also quite a lot of passive monitoring of individuals as well, where state or local public health departments provide information and educational materials to those dairy farm workers, to those dairy farms that do have confirmed outbreaks of H5N1, so that they have information that would allow them to do self-monitoring. And so we understand there's quite a lot of self-monitoring for illness. And from the US CDC's perspective and working with state and local public health departments, we know that there's been more than 220 individuals monitored and more than 30 persons that have been tested for influenza A, with only 1 person having been detected as positive for H5N1 among those more than 220 individuals that have been monitored. Thanks, Wenqing.
Thank you very much, Todd. Todd, would you also be add a little bit about the serology? As there's a question about serology, but not only in animals, but also in cow farmers. So maybe you can address the human part. Thank you.
Yeah, thanks. And so there obviously is a lot of interest in understanding, the potential for seroprevalence studies. Currently, the Influenza Division is working with state and local public health departments to initiate serology studies in farm workers. So we hope to have more information on this very soon. So those studies are being initiated shortly. Thanks.
Wonderful. So with this, we need to move to the question on the animal side now about seroprevalence. And I think this is a question to Dave and maybe also Richard as well about seroprevalence in non-lactating cows. So any serology occurring at the moment in cow population and also in pigs also. Actually, there's a question about surveillance in pigs. So maybe we can start with David and then Richard, if you can have anything to add. David.
Thank you, Wenqing. So to my knowledge, there has not been any data released, but there is ongoing investigations looking at serology, both in the herds that have been affected as well as outside those affected dairy herds, and also looking at serology in beef cattle to see if there has been any exposure to infections there. So currently that is, to my understanding, is ongoing work. Not only the USDA is involved, but also at individual universities and individual state departments of agriculture as they try to get a handle on the epidemiology. There's a fairly large workforce, I know that from USDA and many of their partners with epidemiologists on the ground trying to figure out these issues, and serology is one of the most important components that's being investigated.
Richard, do you have anything to add?
Yeah, I think just add a couple of things. There's some evidence that that at least cows that have I guess cleared these disease, they do produce an antibody response that's detectable by sort of the standard assays. So as David said, there's lots of work going on in the space, but it does appear that at least the assays are fit for purpose in this particular case, which is of course a good thing. I think, Wenqing, you also talked about swine as well. Again, no evidence that swine are involved in this particular instance at all. A number of people have done some experimental infection of pigs with some of these 2344B H5 viruses, with the results sort of ranging from very little replication to reasonable replication, but transmission not being highly robust. So right now pigs don't seem to be playing a role in the 2344B, certainly not in terms of spread at any level. Over.
Thank you.
Richard was talking, I was remembering some. So right now there is the HI is of course our gold standard test for looking for antibodies against the hemagglutinin, but it is not a common test used outside of reference laboratories. But there has been several groups that are looking at validation. I think some of them are actually completed their validation studies of using the ELISAs, various different ELISA tests as a surrogate for the HI test, and those results look pretty good. So it will probably be that most of the serology will be done using ELISA tests as opposed to HI. That will be conducted in cow.
Thank you very much, David. And we look forward to the outcome from these tests. Now that you mentioned about 2344B and this B313, this very specific genotype virus, could you explain a little bit more whether this is the first time detected in US and whether there's anything special associated with this particular genotype? Who would want to start? David.
I can start, and maybe Todd and Richard can add in some information. So the 2344B virus that came across the North Atlantic sometime in 2021 was, of course, at that time a wholly Asian lineage virus with all 8 gene segments. Once it entered North America, it underwent a pretty rapid reassortment, most likely in a variety of wild bird species, with exchange of many internal gene segments with our low pathogenic avian influenza viruses. And there was a paper published by WOAH UK et al. That was in Virology in 2023, is last year. And it outlined pretty well how that reassortment process began in the first parts of the outbreak. And there were multiple genotypes identified. One of those genotypes is B3.2, and that particular genotype in the constellation that it represented then spread throughout North America and became the primary one in North America and also spread into South and Central America. From that B3.2, we get the descendants of the current virus, so the B3.13 is a descendant of that. But also we had a dairy goat infection in Minnesota back in February, and that one was also a descendant of the B3.2, and the genotype is B3.6. But it's a different genotype than the B3.13 that's affected dairy cattle. The one in the dairy goats was a single premise. It was a premise that had infected backyard poultry, presumably that the goats received that virus from that backyard poultry infection, and that within 5 to 9 days of the kidding of the nannies, of the birth, they gave many of the kids died between that 5 and 9 days of age. In this case, that B3.6 virus did not produce infections in the nannies, and there was no detection of that virus in the milk. So it's a different genotype than what's causing the infections on dairy cows.
Thank you very much, David, for this explanation. Let's move to the next set of questions is about milk, about pasteurization. Maybe I'll ask colleagues from US first to introduce a little more about the efforts going on in testing the milk in shelves. And give us some numbers about the percentage, etc., tested. And then I'll invite my colleague Moez to elaborate a little bit about the pasteurization process. Richard, would you want to start?
Yeah, so I can talk a little bit to some of this. So the US FDA has done a larger survey of store purchased milk across the US, and again, maybe Todd has more details on sampling strategy, but from my understanding, this was not a targeted surveillance, so it wasn't just targeting those states that had infected cows. It was more widespread, and they have reported back that about 20% of those are PCR positive for the viral RNA. There's also researchers from the University of Ohio have reported in a smaller sample set, a little bit more targeted states where there have been reports of H5 in cows, and about 40% of those samples were positive for PCR. I also noticed on some social media channels as well, others have posted data. For example, a smaller set focused a bit in New York didn't identify any PCR positive. So it does seem that the positivity may depend a little bit on where sampling is done. I can say that we've tested in excess of 50 of the sort of lowest CT values, meaning those samples that have the higher RNA load. And by putting those samples into both embryonated chicken eggs and also cultured MCK cells, we have not been able to identify any viable virus in those samples. So there has been a relatively large number of samples tested, but so far from a safety perspective, it does look like the pasteurization process is removing viable virus from those samples. Okay.
Thank you, Richard. And I would like to invite my colleague Moez to comment about the pasteurization. And if you could add something related to cheese making as well. There are also questions about cheese.
Yeah, thank you. I will repeat my statement that for the moment, all the evidence that we collect are in favor that pasteurization is inactivating the virus for the moment. I think the question about cheese is very important. If there is pasteurization, and if there is no pasteurization, and I think a lot of questions I saw them about cheese made from raw milk. I think it's very important to understand that we have a huge variety of cheeses with different technologies, and in such situation we need to think on harder approach. There is no single action that will solve everything. There is different barriers that can be built from the farm level, looking at sick animals, deviate milk from sick animals. You have the temperature, the heat, the possible heating, the lactic acid during the fermentation. So we need to think on all those barriers and end up with a sort of risk assessment, risk-based approach to understand if the process is safe or not. However, in the Codex Alimentarius, we have international standards that deal with pasteurized and non-pasteurized milk products. And there is a lot of recommendation in the same approach, harder approach, that will provide at the end the safety of the products. However, when we are faced to new type of contamination, it depends on the food safety authority. It's a risk management decision that can be made by the food safety authorities is to stop, halt for a certain period of time the production of cheeses with raw milk, waiting for better understanding of the safety of those products and to understand if the technology in place is enough to produce safe products. Over to you.
Thank you very much, Moez. Okay, let's move on. So I'll ask Magdi to address a question about the WHO surveillance on H5N1. There were also questions about wastewater surveillance, etc. Can you comment on that, Magdi?
Sure, Wenqing, thank you. This is an important question. The WHO Global Influenza Program provides member states with a lot of strategic guidance, technical support, and coordination of activities that are essential to make the health system better prepared against seasonal and zoonotic and pandemic influenza threats to populations and individuals. So in terms of the surveillance for zoonotic influenza, the National Influenza Centers, which is around 152 in 130 member states of WHO countries, they conduct routine ILI and SARI surveillance every year, and part of that they also have the capacity to detect not only seasonal influenza but also zoonotic influenza viruses like H5N1 and H9N2 and others. And through this network of the WHO, the GISRIS network, or the Global Influenza Surveillance and Response System, they detect these viruses. And according to the ICHAR, International Health Regulations, when they confirm by lab testing any of these zoonotic infections, they report those cases to WHO via the official channels from the member states. And those cases now, after reporting, they are handled on the national level, the regional level, and an HQ to make sure that the virus does not transmit further. There is contact tracing and further characterization of these viruses through sharing with the collaborating centers, as mentioned in the presentations, in order to select new candidate vaccine viruses. Sujovirus has evolved or changed from the currently listed CVVs.
Thank you very much, Maggie. I think we are already on time here now. About the wastewater, just to add that certainly wastewater surveillance is one of the surveillance methodology, especially in recent years, has been developed in many parts of the world. But for influenza, for the routine influenza as well as for the outbreak investigation, certainly like in the localized areas, in farm, using wastewater surveillance as complementary approach to the syndromic surveillance certainly would be very valuable for those. A lot of questions still there. We'll discuss with the EPI-WIN webinar team to see whether we will be able to provide written answers to you, but we'll explore if it's possible. Certainly, we'll do so. I was told that Maria would want to also say some words at the very end. She's still connected. Very good. So just to say, colleagues, that H5N1, it's not just in US. As many panelists already said, it emerged in 1996, and since 2002, it's been endemic or spreading globally, but it's the first time in cows. So this is the reason where WHO are concerned about this situation, are watching that closely. So we'll be continuing working with you all in all the member states on the front of the surveillance and preparedness for H5N1, basically, or also for other influenza as well. So with this, I pass over to Maria to conclude this session.
Thanks so much, Wenqing. Just wanted to reiterate the points that you've just made. The focus today has been a lot on what we're seeing that's happening in the US, but there's this epizootic that we've been tracking for a few decades now. And we're not sitting back and monitoring the situation, watching the situation. There's a lot of active surveillance that's ongoing in different animal species. We're asking for more, particularly that we've seen expansion into dairy cattle. But this is a global partnership, and we need help from all of our member states. We need help across the different sectors. We need help from scientists, public health professionals, animal health professionals to be able to assess each of these viruses. So it's a group effort. And we're really grateful for all the partners who have worked on this for decades, and ask them to continue to work with us as we go into the future. So thank you very much. There's a lot of activity in the Q&A. We've answered about 65 questions on my count. We didn't get to all of them, at least written, not to mention the panel itself, which I think is pretty good, Supriya. You could tell me if I'm wrong. But we're here to answer more questions, and we will use all of the different platforms that we have, whether it's through EPI-WIN, whether it's through our regular press conferences, but also in other channels that we have. And with that, I'm going to pass it back to you, Supriya, and then all of us are going to go back to work. Back to you, Supriya.
Thanks very much, Maria, and thanks to Wenqing and all the panelists for this fantastic webinar. I think we've had more than 130 questions, which indicates the sheer level of interest in this topic, and it's been really very interesting. I'd also like to thank everyone who's worked on this webinar in the background, my colleague Sadrish Pradhan, who's been working on the chat and monitoring all the questions as well. Thank you very much. Special thanks to Aspen for all her hard work in organizing this webinar. Really appreciate it. And we look forward to seeing you at the next webinar. I hope we will have as much of an attendance as we did today. So our next webinar is next Thursday at 1 o'clock. It's on health systems for health security. And we have fantastic participants on EPI-WIN. We'd love to know how you use this information. If you use it for your clinical practice, if you're an academic who uses it to update your courses, if you use it for workshops and trainings and capacity buildings, or if you're in government and use it for policies and plans, please do let us know. Share your documents, your policy papers, your photos, your links, and write to us at EPI-WIN. And thank you so much for joining today in this fantastic webinar. And once again, thanks to all the speakers, all the panelists, and particularly to the Global Influenza Program. Thank you very much, and goodbye.